Good evening, everyone. My topic that I have chosen is the effect of honey, high-frequency wanding, and LED light therapy on Staphylococcus epidermidis, an acne-causing bacteria. For a little information about myself, my name is John Mills. I'm a senior at Tunstall High School, and in the fall of 2023, I will be attending East Carolina University, where I will be pursuing a double major in biology and chemistry. There may be questions as to why I've chosen the topic that I have. This topic struck particular interest to me because I've had problems with my skin growing up. I've had pretty severe atopic dermatitis, also known as eczema, all my life. However, my interest for my career goal of becoming a dermatologist really took off when I contracted a bacterial skin infection at the age of 13. This infection greatly affected my health and wreaked havoc on my skin. Additionally, while conducting research on treatments for acne, there was a significant lack of research on the treatments that I have chosen and treatments for Staphylococcus epidermidis. Acne vulgaris is a very common skin disease, more often referred to as just acne. This disease presents differently in every person and is usually seen as comedones, papules, pustules, virulent cysts, and even extends to the scars as a result of these. Acne is not limited to the face and can actually occur on any seborrheic area of the body. This means that anywhere you have pores, you can get acne, which means everywhere on your skin. Staphylococcus epidermidis is a gram-positive bacteria that is responsible for causing pus and inflammation where an infection is present. This is important to acne because it is a contributor to the actual pimples that are formed. If there is no inflammation, then there are no pimples. The traditional approach to treating acne are the standard treatments that are received when going to a dermatologist. There are many treatment options that dermatologists can choose from, which include oral treatments like Accutane, topical treatments like medicated ointments and creams, face washes, and many more. However, despite there being plenty of treatment options, research has shown that there is still a need for future treatment options in order to reduce side effects like photosensitivity and antibiotic resistance. Additionally, topical treatments are most commonly used as a first resort when treating acne. And a study comparing different topical treatments discovered that the effectiveness of different topical treatments determined that a combination of adapalene and benzoyl peroxide was the most effective topical treatment combination. However, it's still not optimal. This is where the solution to this problem can be introduced. Non-traditional treatments can be described as treatments that skew away from what was mentioned previously and serves as alternatives to them. For example, a study regarding therapeutic fasting paired with yoga modalities was conducted and showed that a combination of these two therapies significantly improved the symptoms of acne vulgaris. Another type of alternative medicine that has been studied is light therapy. A study was conducted on the effects of blue light therapy on people who have acne, and the results yielded suggested that it is effective at killing the bacteria Propionibacterium acnes, another acne-causing bacteria. Similar to light therapy, a study was conducted on the effects of laser therapy on people presenting with acne. This study also yielded results that suggest that CO2 laser therapy is a viable option. And finally, there was a study conducted on the antibacterial effects of Manuka honey on Clostridium difficile, also known as C. difficile. The Manuka honey proved to inhibit the growth of C. difficile. All of these studies show the relative efficacy of alternative treatments as viable options, but none of them mention Staphylococcus epidermidis or the treatments that I have chosen on Staphylococcus epidermidis. To verify this trend, a scientometric review, which is a type of analysis used to physically map knowledge in one area, was conducted and showed that there was a significant lack of research on both alternative medicine and dermatology as a whole field. This is seen through another review conducted to compare alternative treatments. This review repeatedly stated that the studies in the review were either faulty or not backed up by other research to be more reliable. All of this leads to the importance of my research and highlights the gap within the field that I hope to bridge. This lack of research has led me to my research question, which states, which treatment between Manuka honey, high-frequency wanding, and LED light therapy is the most effective at reducing the growth of Staphylococcus epidermidis? The hypothesis I formed in relation to my research question was that if the non-traditional treatments of honey, high-frequency wanding, and LED light therapy are applied to Staphylococcus epidermidis, then the honey will have the greatest effect at reducing the bacterial growth. And my null hypothesis is if the non-traditional treatments of high-frequency wanding, honey, and LED light therapy are applied to Staphylococcus epidermidis, then none of the treatments will show a significant amount of reduction in Staphylococcus epidermidis over the others. The independent variables for my experiment were the different treatments that I applied. The dependent variable was the bacterial growth. My controls were Petri dishes cultured with bacteria that received no treatments. And my constants were the temperature and environment of the Staphylococcus epidermidis, a clean working space that was disinfected. I kept the length of treatments per group the same, and I also did all the treatments as close together as possible. I used the same brand, type, and amount of honey. I used the same brightness and length of LED light, and I used the same high-frequency wand for each dish. And the method of growing the bacteria was all the same. Now on to my actual experiment. First, I gathered my materials, which included 11 Petri dishes per treatment group being tested. And specifically for the honey treatment, I used an agar medium that had the Manuka honey mixed in with it. I then collected an inoculation loop, Bunsen burner, and the treatment being tested. After I gathered my materials, I then inoculated the bacteria onto the Petri dishes, being sure to keep everything as sterile as possible. Once all the dishes were inoculated, I then applied the treatment. The honey treatment was the dish itself because it had the Manuka honey infused with the agar. The blue light therapy was 20 minutes under the blue light in a mostly enclosed chamber. High-frequency wanding was 1 minute of wanding per dish. After treatment was finished, I then put the dishes into the incubator at 37 degrees Celsius. After 24 hours had passed, I checked the dishes for growth. Then I collected the data, making a percentage comparison of the growth of the bacteria on the control and treated dishes. I then performed my statistical test. The statistical test I performed to interpret the data I collected were a one-way ANOVA and two-sample t-test. The one-way ANOVA yielded a p-value that was less than 0.001. This allowed me to compare the mean coverage of all the treatment groups to determine whether or not my results were statistically significantly different. Because the p-value was less than the confidence interval of 0.05, this allowed me to reject my null hypothesis. The two-sample t-test allowed me to determine whether there was a statistical significant difference between each of the groups separately. All proved to be significantly different because the p-value determined was less than the confidence interval of 0.05, allowing me to reject my null hypothesis. Here I have created a bar graph with error bars, which are circled, to display my statistical test results. The error bars are the important aspect of the graph in relation to my statistical test because they all show significance. I calculated the standard error for each test group and then created the error bars. Since the error bars do not overlap, that verifies that my results are statistically significant. Also, it is important to bring attention to the mean coverage for each group, as this will be important to the concluding factors of my results. Honey had a mean coverage of 0%. LED light had a mean coverage of about 10%, and the high-frequency wanding had a mean coverage of about 40%. With all the statistical factors accounted for, there is sufficient evidence to conclude that Manuka honey had the most success at inhibiting the growth of Staphylococcus epidermidis at a confidence interval of 0.05. This can be further supported by the fact that the mean coverage of growth for the Manuka honey group was 0%, when the other two groups had visible growth. The fact that there was no growth for the Manuka honey group is a very simple and tangible piece of evidence that supports my hypothesis that honey was the most effective. The reason that Manuka honey performed the best was because of the contents of the honey. The contents that I explained its special properties would be its low pH, high sugar content, which dehydrates and kills bacteria, and the antimicrobial agent of hydrogen peroxide found within Manuka honey. This is backed by the study that was mentioned in my review of the literature on Manuka honey, which also showed a complete inhibition of bacterial growth. Additionally, my findings in regard to LED light therapy were also consistent with the results explained in the literature review. With my results explained in the literature review covering the effects of blue light therapy on bacteria. High-frequency wanding worked the least, but since there was no literature available on the effectiveness of high-frequency wanding on the inhibition of bacterial growth, there is still no baseline to compare it to. The limitations to my study include the accessibility to a completely representative sample to cover all acne types and bacteria. This limitation exists because I did not have the ability to confidently perform my test on humans. Also, the fact that the different groups require different methods of administering treatment means that there could be some error due to human error or lack of efficacy because of the way I administered the treatment. Lastly, even though my sample size was 30, I still consider this to be a small sample size, which can limit how accurately my results represent the population. I have found that Manuka honey proofs have extremely effective antibacterial effects on Staphylococcus epidermidis, but future research should aim to test if it has the same effect on other bacteria, especially those associated with acne. Also, there could be a possibility that there are other honeys that have similar or the same effects as an antimicrobial agent, and further research could solidify that. While my research displayed a lot of information on the positive effects of honey because of how well it performed, there is still not much knowledge on the benefits of LED light therapy and high-frequency wanding. While these may not be the best options for treating Staphylococcus epidermidis, they could prove to have some other great effects that were not within the scope of my research. There was lots of trial and error throughout every step of my experiment, from the formation of my research question to the interpretation of my results. From this, I learned that failure was part of the research process. And with each failure, I learned something which helped me improve my experiment and learn more about research in general. For the conclusion of my presentation, I would like to give thanks to those who helped me along this laborious process. I would like to thank Dr. Bocock and Dr. Jones for approving my experiment. And last but most certainly not least, I would like to thank Ms. Dillon and Ms. Long. Ms. Dillon was so much help when it came to formulating my experiment and actually performing it with the correct laboratory and biological techniques. And Ms. Long was the main person behind all of the written parts of my experiment and helped the best she could with statistics and in the lab. At this time, I will open the floor to any questions. Ms. Davis.
So, if I'm understanding correctly, you inoculated your plates, like the whole plate, and then you treated them, and then you were looking for percent coverage, like of a lawn of bacteria. You weren't counting colonies or anything. Is that correct?
So Ms. Davis is asking about my procedure for my experiment when testing the different treatments. And the way I did it was I inoculated the bacteria all over the dish and administered the treatment like you said. And then after 24 hours had passed, I checked the dishes for growth, but there were no separated colonies and there was nothing I could specifically count. So what I determined I needed to do was take a percentage comparison, and that way I could compare the groups without having a specific number of counted colonies because they weren't grown in specific colonies. Did that answer your question?
Absolutely. Yeah, thanks.
Ms. Probst.
In your literature review, as far as human trials, did you find a specific age where honey cannot be used? The reason I'm asking is infants, usually they caution you not to use honey with infants because of botulism toxin. So in real life, would there be an age limit to using the honey?
Ms. Probst is asking about whether or not there would be an age limit to using honey as a treatment. And in my literature review, I did not find anything that stated specifically anything about age restrictions. And specifically for this topic, I don't think it would necessarily be applicable because it's being used as an acne treatment. And I'm not sure if there's any illnesses that are associated with acne in infants.